While planning a microarray experiment, like any other experiment, there are some experimental design considerations, which are like, for example, how many replicates we should use in our experiment. We need to amplify the RNA because sometimes we have RNA concentration is very low, or maybe if we are using a small number of cells, we can amplify the RNAs, and since every RNA is amplified, it's okay to do these PCR kind of things. Sometimes we do these dye swaps also, so that if we have any biasness for the binding to specific dyes, that must be removed. Sometimes we also use a reference sample where we compare the expression of different samples with that particular reference. Replicates, they reduce the variability in summary statistics, and they allow us to perform some formal statistical tests. Remember guys, for statistical tests, you cannot compare two numbers, you need two distributions, and for a distribution to be there, you need a mean and a standard deviation. So at least two replicates are important in order to make it or treat it, your experiment as a distribution. Whereas Lee et al. Has recommended that there should be at least 3 replicates. So replicates are kind of divided into two major components. One of them is technical replicates, and the other kind of replicates are biological replicates. Technical replicates are repeat hybridizations using same RNA isolate. So you are repeating your experiments with most of your things in your experiments are the same. So for example, sometimes you take different readings from the same organisms, maybe a plant or an animal. In that case, these readings are taken over the same individual, so maybe we can classify them as technical replicates. So you have less variability in your data sets. Whereas biological replicates, they use RNA isolated from separate experiments or some separate organisms. We can have, for example, we are extracting our RNA from 2 different humans or 2 different plants. So in that case, they are biological replicates. Though technical replicates can be useful for reducing variation due to hybridization or imaging. So in here, we can reduce the issues where we can have some variability due to hybridization or imaging in your experiment. So your experimental procedures, their variations, we can reduce them, but biological replicates are necessary for a properly controlled experiment. So amplification methods, as I said earlier, they are there in order to amplify your RNA so that you can capture some low expressed genes, and you can also start with small number of cells. While we are using 2-color arrays, the dyes, we are using green and red, sometimes there is biasness for specific kind of samples to have their interaction with different dyes is different. So in that case, there must be some biasnesses. So in order to reduce these biasnesses, we swap the dyes. So we take one sample, for example here, now we take sample 1 and we give him red color, we attach it to this red dye, and then sample 2 is labeled with green, but in a second round we will switch these dyes. So sample 1 is green now and sample 2 is red now. So this dye switching or swapping helps us to reduce the biasness in hybridization with these different colors. Sometimes we use reference sample in one of the channel of the arrays. This will usually something that will hybridize to most of the features, maybe a complex RNA mixture. So that is, we put it as a reference. So using a reference, it allows different comparisons to be made between different experimental conditions as each is compared to the common reference. So here we have, for example, reference samples. So each sample is sample 1 and 2 and 3 is compared with the reference. So S1 versus R, S2 versus R, and S3 versus R. So reference is same for everybody. So in this way we can standardize our procedures. Our results, they must be validated. So microarray results, since we are going on a large number of genes, but sometimes we are interested in a few set of genes. So for that we might design some molecular experiments where we can validate our results. For example, we can do these RT-PCR kind of things for some of our genes. So we need to validate our results. We might want to use northern blots, we might want to use QRT-PCRs, etc. It's always good to have two kind of separate pieces of evidence in order to support our results. Miami, or minimal information about a microarray experiment. We have talked about this thing before when we were discussing the GEO, gene expression omnibus database. So over there we see that in order to publish a microarray experiment, it is expected to make some available information. Now we call it as a minimal information. So this is what minimum which is required. So it allows others to evaluate the data and compare it to other experimental results. So we need to talk about the experimental design. How many factors were there? What type of experiment was there? How many arrays they were used? Reference samples, if anyone was used, and how we performed quality control. And here we can also put the database accession numbers, maybe GEO or array express. So those kind of things, they are part of experimental design in Miami. We talk about sample used, preparation, and labeling. We need to provide information about hybridization procedures and different parameters that were used. We need to talk about measurement and specifications like quantifications, hardware and software used for scanning and analysis. How did we perform our raw measurements? How did we do our transformations or normalizations, etc. We also talk about array design, which platform was used, how many features and their locations, and what manufacturing protocols we used. So here we see that in order to set an experimental design, we should consider some important factors like how many replicates are there, whether we are amplifying our RNA or we are not. We need to do dye swaps. Sometimes we need to put the reference in our experiment, and we need to capture all these information and provide some minimal information about our array experiment.